Hey there, students. Today I'm going to be giving a little bit of an introduction to what we're going to be doing in class. It's a case study of sorts, or a hypothetical case study. It's not really real. That about two different people named Chucky and Melody, or Charles, who goes by Chucky, and Melody, who travel to a resort in Colombia to be able to experience different types of things. Um, in general, the whole entire story is a little bit hokey, but it is what it is. And it's also all written just so that you can answer the questions about it. Those questions are going to have to be turned in on Canvas by taking the quiz thing that we put together. For all of it, I want you all to review the neuromuscular junction. I'll do that in just a second. And then there's also a couple other different questions about three different chemicals. Well, four, but really three. One of them is a chemical called curare or curare, but it's curare. It's a plant-derived type of chemical that can be found used in a large number of different types of Amazonian cultures to put on the tip of a dart to make it so that it can be used for hunting. There's also botulinum toxin, which is well known as the therapeutic drug Botox. Botulinum toxin is actually fairly common within, or it used to be common within the food industry. It's derived from a bacteria called Clostridium botulinum. It would infect cans. And then if you happen to end up eating the food in that, then you would have terrible issues that would happen. But now it's been found to be fairly effective at causing paralysis of specific muscles so that people's faces don't turn wrinkly, or it's a common way of trying to treat migraines by releasing some of the neck muscles. And by releasing, I mean paralyzing so that you can't actually use them. But if they're the things that are causing some sort of migraine or tension headaches, then that's how you do it. The third, which is actually the second, but the third and fourth, the other chemical that we're going to be talking about is a nerve gas called sarin gas. There's a slightly less terrible type of derivative of that that's called physostigmine. And there's also another version of that that's called neostigmine, but both of those are essentially set up to counteract a variety of different other types of things. So if you are looking for something to stop paralysis, it causes a different type of paralysis rather than flaccid paralysis where you can't move anything. It causes something that's more called turgid paralysis, where you're stuck in a single position. So it can be used as a therapeutic when you have different types of muscle relaxants. So it's the opposite of a muscle relaxant. So those are the three things. We're going to look at how the actual biochemical ways that they work and how they might end up working, but I'll talk about that when I'm over at the board. Okay, so I know that there's a lot of videos that you can watch of this, but I'm going to talk through this pretty quickly just so that we can get to a couple other different things later on. But one way or the other, as we're going through this, just remember that you can watch this over and over. So this whole thing is all going to be dealing with the neuromuscular junction. So we have to have our motor neuron, which is the thing that's at the top, and then also our muscle, which is the thing at the bottom. Obviously, I'm going to try to draw in a couple different channels and some other fun things like that. But the synaptic tethering proteins are actually going to be fairly important for this, or they're also called SNP proteins or snare proteins. But they are the squiggly lines that are holding on to the little egg-shaped things that I have. And inside those eggs, we have acetylcholine. Otherwise, we also have a couple other channels. Most of my channels are going to just be kind of like straight lines or something else like that, but it's something else to keep in mind as we go forward. Then we have an action potential that's going to come down the axon of the motor neuron. While that is happening, then I'm just going to draw that as a line. An action potential, remember, that's a depolarization, so that's going to cause voltage-gated calcium channels to open. Once those calcium channels open, the calcium goes to the synaptic tethering proteins. The synaptic tethering proteins are going to shorten and then that's going to release the acetylcholine to the cleft. Acetylcholine is going to bind to the nicotinic acetylcholine receptor and then cause it to open. You can see me doing a variety of different types of things that say, hey, look, it's opening. Ooh, it's opening. And then that is going to cause the opening of the next voltage-gated channel, which will cause the next voltage-gated sodium channel. Remember, sodium is going to open before potassium. The potassium channels are a little bit slower, and then potassium is going to follow its electrochemical gradient and leave, and that's going to continue all the way along the sarcolemma until it goes into the T-tubules. Once again, sodium is going to be causing the depolarization, potassium is going to cause the repolarization, and resting membrane potential brings us back to normal. The acetylcholine then, once it's done its job, it's going to hang out in the cleft until it either dissipates and just goes away or gets broken up into acetic acid and choline by the acetylcholinesterase, that's the Pac-Man looking thing, or it can be taken up again by the cell. I don't like the word reuptaken, or it sounds awkward, so taken up by the cell again. Now we'll talk a little bit about what happens with Botox and curare and sarin. Okay, so here I am again as a floating head this time instead of, the back of me with a disembodied voice or actually just, talking to me. But one way or the other, here is a visual or an example that I actually made in PowerPoint of this exact process, kind of like I drew out on the board. It's just a little bit cleaner and easier to see things. When you are looking at this, then one of the things that we want you to think about is where these different types of toxins are going to take place and where they're going to fit on this whole entire thing. Because yeah, this is really cool if we end up talking about it, but really if everything breaks down, then it's a whole lot more fun to be able to talk about things like how can this break down. Well, obviously the voltage gated channels can stop working, the nicotinic acetylcholine receptor can stop working, the synaptic vesicles cannot release their stuff. It's fun things like that make it so that it's a little bit easier to play around with these things and learn them. So for this, we're going to look at how Botox works, how botulinum toxin works, essentially, and what it does, where it screws things up, where curare screws things up, and where sarin screws things up. And really, you can look at a variety of different types of neuromuscular toxins, and they're going to do similar things across the whole entire board. You can find them in cone snails, for example. It's a really cool type of snail that causes paralysis. But this is what I want you all to look at. This is what I kind of want you to kind of keep in mind when we're working through this whole project. Okay? Have fun, good luck, and if you have any questions, please just ask.